Hello, today I'm going to talk about the influence of phytosterols and ergosterol on wild alcoholic fermentation for Saccharomyces cerevisiae strains. Sterols are lipid compounds that are responsible for ensuring yeast growth, metabolism, and viability, and also to resist to ethanol stress during wine fermentation. Ergosterol are synthesized by yeast in the presence of oxygen, while phytosterols are assimilated from solid particles from grape must. Until today, there is no knowledge about the physiological impact of the assimilation of phytosterols and ergosterol. And that's why I'm going to present you today our study that compares the performance of a wide set of Saccharomyces cerevisiae strains fermenting with phytosterols or ergosterol under two conditions: sterol limitation, mimicking excessive mass clarification, and high sugar content that leads to ethanol stress. In order to mimic sterol limitation, we worked with a synthetic must that had a low content in ergosterol or phytosterol. To mimic high sugar content, we worked with a synthetic must that had a low concentration of sugars. We tested 27 Saccharomyces cerevisiae strains, and fermentations were done with the robot fermentation that allowed us to follow fermentation kinetics and also to sampling. Results for sterol limitation showed us that strains that were able to assimilate more amino acids were also able to produce more biomass and then to achieve a higher maximum fermentation rate. And then these strains were able to start fermentation faster. Moreover, we can observe that strains that had a higher viability at the end of fermentation could finish fermentation early. Increasing, there were no correlation between the start of fermentation and its achievement. We can also observe the huge phenotype diversity of strains tested in function of the sterol type. ANOVA result showed us that it was phytosterols that allowed a faster fermentation, and then it was ergosterol that allowed a rapid achievement of fermentation thanks to a better viability at the end of fermentation. Regarding high sugar content results, we can observe the same correlations between variables as seen from sterol limitation, and differently from seen for sterol limitation, it was ergosterol that allowed a faster start of fermentation, and as seen from sterol limitation, it was also ergosterol that allowed to finish fermentation faster. To conclude, this study highlighted the importance of taking into account the sterol type available during wine fermentation. And the most remarkable result was that under both conditions, it was ergosterol that allowed to finish fermentation early, thanks to a better viability at the end of fermentation. Moreover, we showed a huge phenotype diversity of Saccharomyces cerevisiae strains. Thank you for listening to me, and don't hesitate to contact me if you have questions. Thank you.